Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Okay, hello everybody, welcome back to the A-Level Cookbook. Today we're going to be doing the OCR A-Level Biology Paper 1, OCR A from 2021. So which of these is not a function of the liver? So it says production of urea in the ornithine cycle. So that is what happens. In the liver, what it does is it removes any amine groups, NH2s, from like amino acids. But the problem is that would make ammonia, which is toxic. So then what happens is the ammonia is toxic. So what happens is we combine that with CO2 in the ornithine cycle, and that creates urea. So what happens is ammonia and CO2 gets combined into forming carbamoyl phosphate, which becomes citrulline, and then all of a sudden it, whatever, a lot of other stuff happens. But basically, that one's out. So then it says removal of amino acid groups in the amino acids. Well, deamination is what that is, and that does happen in the liver. Next. Storage of excess amino acids and proteins. No, it does not. So that one's it. And glucose as glycogen is stored in the liver, so it is that. So C is correct. So which of these statements about the control of blood glucose is correct? So pancreas cells increase their release of glucagon, the blood glucose concentration rises above it. Well, no, because remember that glucagon elevates the blood glucose concentration, so it only do it if it was too low. Next. Glucagon stimulates the conversion of glycogen into glucose. Well, yeah, because that's the storage molecule for glucose, and converting into glucose means you have more of it. So that one's out. That one's correct. What else have we got? Insulin increases blood glucose concentration, but no, it doesn't. It decreases it. The interaction of insulin and glucagon keeps the blood glucose concentration constant. Well, not necessarily. There's lots of other factors that come into play as well, and also it's always a wiggle in between them. So, no. So it's B. Next, it says, which of the following options describes the path taken by proteins such as digestive enzymes when they're exported by a cell? Golgi apparatus to rough endoplasmic reticulum to secrete a vesicle doesn't make sense because the proteins are made at the rough endoplasmic reticulum where the ribosomes are, where protein synthesis happens, so that one's out. Ribosome is where protein synthesis happens, that adds up. A smooth endoplasmic reticulum synthesizes and processes lipids, and it doesn't have any ribosomes, so that one's out. Rough endoplasmic reticulum has ribosomes, and the endoplasmic reticulum can also fold and coil and help things like, fold and process proteins, so that adds up. Golgi apparatus can package them, that adds up. And then secretory vesicles, well, that means that they can actually be, packaged and move across to wherever they need to be, so that actually adds up. Smooth endoplasmic reticulum, again, is lipids, so nope, so it's C. Next, then, it says which of the statements about enzyme catalyzed reactions is true. So binding of a substrate to the active site can weaken bonds. That is true. Enzymes allow reactions to happen at a rate faster than Vmax. Well, no, because the Vmax is only at the certain point at which the enzymes can maximize the rates until something else becomes a problem. So, for example, I could have all the enzyme in the world and all the substrate in the world, but they can only peak. The other factors, so for example, like temperature, pressure, pH, and stuff, will probably limit the reaction, and I have to tweak those. So that one's out. Enzymes increase the rate of energy by increasing activation energy. No, they don't. They lower it, and then every 10 centimeter increase in temperature will approximately double the rate of that. Well, no, that we don't know that. It can vary. So it's got to be A. So now it says the figure shows the metabolic pathway leading to the synthesis of 5 amino acids, aspartate, lysine, methionine, threonine, and isoleucine. So E1 and E8 is whatever, right? So it's different enzymes. And it says which of the options is an example of an end product inhibition? Okay, so the end of this metabolic pathway, what could happen is that this final product can inhibit the enzyme that stops the original reaction and causes the entire thing to stop. So what we need to do is we need to go through each one of these options and see what they could be. So aspartate inhibits A3 and A4. Well, that's not the end. That's not an end product. So because that would be what here. So that's out. Isoleucine inhibits E7. Well, if it inhibits E7, then it just means you can't do that. So that one's out. Lysine inhibits E1 and E8. Well, if you inhibit E1, then that can't start. So that could be it. And then methionine inhibits E8. Well, no, because then you just wouldn't get that. So it's got to be C. So the question says, here's some adaptations to specialized cells. So biconcave with flatter nucleus. Nope, they don't have a nucleus. Biconcave with no nucleus could be possible. Biconcave with no nucleus could be possible. Biconcave with flatter nucleus. No, that one's out. So then neutrophils, large spherical nucleus. Nope, because neutrophils have multi-lobed nuclei. So that one's out. Large spherical nucleus. That one's also out. So it must mean that it is B. And there you go. But even if we went further, well, the multi-nucleus thing does let them squeeze through smaller gaps, so that's correct. And then palisade cells and thin walls allowing rapid diffusion of carbon dioxide. Yes. Thick cell walls. No, because these are these are palisade cells. Remember, so this is where all the gas exchangey stuff happens. So it's not that the thick cell walls maintain the turgor. It is that fact that they have really big vacuoles in them, which keeps the cell turgor to maintain turgor pressure. So the graph represents the potential difference across the membrane of an axon during an action potential. Which position correctly describes the events in action potential? So whenever you've got nerve impulses, what happens is, just as a quick crash course, sodium ions enter, right? So you've got sodium ions entering here. That could be like whatever because of your generator potential. When the threshold is hit, what happens is voltage-gated sodium ion channels open, which means now this membrane can have loads of sodium coming in. So sodium enters, but then it stops. Now the way I like to think of it, to make it a bit easier, is that if it was just you know, if these channels are open, it would just continue shooting up like that. So something's stopping sodium moving in, because depolarization means sodium entering. So what that must mean is that at this point when it stops going up, the Na plus channels have to close here, right? But now it's going down. So something else has to make it become more negative, meaning that we have to lose positives out from inside the cell membrane. So if we open sodium ion channels again, it would just shoot up because sodium wants to move in. So it's got to be that the potassium ion channels must open here, so K plus can leak out. So K plus moves out via effusion, and then what happens? We overshoot here. So then if we're overshooting, it means we're losing too much K plus. What happens is that the K plus channels here are too slow to close, so we overshoot, and then we're back to normal. And that makes sense, because in your normal resting membrane potential, you're pumping out 3 sodiums for every 2 K plus in. And also, as well as that, K plus can freely just move out as well, just a couple of them. The reason is, well, again, the way I like to make sense of it is that this sodium-potassium pump will not work if there's no potassium here. So to keep the pump going, a little bit of potassium can leak out, but sodium can't come back in when you're in your resting membrane potential. And the way I like to think of it is that because we're shoving so much sodium out, the concentration of sodium outside the nerve is really high, and the concentration of potassium inside the nerve is high. So when you finally open those voltage-gated sodium ion channels, because the concentration gradient is so unbelievably high here compared to this, they just want to shoot in like there's no tomorrow. And that's why you get that sharp increase. But then if we kept it open, it would just keep entering and entering and entering and entering. So something must have happened to stop them entering like that, right? But now we need to get this back down to normal, because at the resting membrane potential, we put so much potassium in us, the potassium ion channels open, so it can leak out, and that gives you that negative there. So now it says which statement about the graph is correct. So first one says depolarization is happening at 4 and hyperpolarization is happening at 6. That is incorrect, because all of it going down is repolarizing, bringing back that negative inside versus out, so bringing back the pole. Depolarization is happening at 2 and then hyperpolarization is happening at 5 is correct. Hyperpolarization is happening at 5 and repolarization is at 6. No, it's not. Repolarization is occurring at 4 and hyperpolarization is happening at 6. Well, hyperpolarization is where you overshoot, and that's not that either, so out. Okay, so this figure shows the results of 3 pregnancy test strips with mobile fixed immobilized monoclonal antibodies binding to human chorionic gonadotrophin. The mobile monoclonal antibodies are labeled with colored beads. The results on the control line contain the fixed monoclonal antibodies. Each strip is dipped in a sample of urine up to stop line and then laid for 10 minutes in red. So here we've got our control line, our results line. Remember, it's got fixed monoclonal antibodies here. And we have our stop line as well, and that's fine. So remember that the question tells you that when you put a urine on it, if there's human chorionic gonadotrophin in it, it will bind to it'll bind to the mobile monoclonal antibodies, which then bind to the fixed monoclonal antibodies, and it'll give you either a positive or a negative. Now the control is there just so that you have it as a comparator, so it's always going to be like that. Anyways, so it says which of the statement about these pregnancies tests is true. So in strip 1, HCG is bound to mobile antibodies that are captured by fixed antibodies on the control line. The pregnancy test is positive. No, because that's the control. It's always going to be there, so you know what to compare it to. If there was HCG, then it would bind to those mobile antibodies, which then bind to the fixed antibodies here, and you get 2 strips, so that one's out. In strip 2, HCG is bound to the mobile antibodies. These have been captured by fixed antibodies. The result sign this pregnancy test is positive. That is true, because again, the control is always going to be there. If the HCG is there, it binds to those antibodies that are mobile, and then it binds to these fixed ones there, so that one is correct. In 3, it's bound to mobile antibodies. These have been captured by the fixed antibodies, and the result sign the pregnancy test is negative. Well, no, the control line should be there, so we can't really tell if it's positive or negative. Out. In strip 3, the HCG is bound to mobile antibodies, and these have been captured by fixed antibodies. The result line is positive, so, the results line is... So again, if it was HCG was here, bind to antibodies, and then these are mobile and bind to these ones, it would be positive, but we don't have any control, so that one's out, so it's B. Next, it says below is the diagram of a neuron. The arrows show the impulses of the direction of the nerve impulse. We've got 7 and 8. So it says which table correctly identifies structure 7 and 8 and the type of neuron in this diagram. So below is a diagram of a neuron. The arrows show the direction of the nerve impulses. So we can see it's starting neuron, it's going that way, whatever, right? It says which one correctly identifies 7 and 8 and the type of neurons shown in this diagram. So remember that, dendrons will carry impulses from whatever it's coming from to the cell body, and then axons take impulses from the cell body to wherever it's going. So number 7 is not an axon, and so number 7 is a dendron, number 8 is the axon. So those two are out straight away. Now if we look, we've got dendron, axon, that's fine, but we need to decide whether it's sensory or motor. So remember, motor neurons, they get impulses from whatever it could be. So it could be a relay neuron or a sensory neuron, and they have one long axon and lots of short dendrites, right? sensory neurons, what they do is they have one dendron, which carries the impulse to the sensory cell body, and then one axon, which takes them away from the cell body. So by definition, this one has to be sensory, which is C. So if I were to draw that as a diagram, this is going to be a really rubbish diagram. We've got the dendron, cell body, and then the axon, and then wherever it's going here, right? That's sensory. A motor one would look like this. So you've got your cell body here with lots and lots of dendrites coming off of it. You've got just one singular axon, and it goes to wherever it needs to be. This one here has a dendron and an axon, so it's sensory. So then it says, which of these following statements about nervous transmission is not correct? So breakdown of myelin sheath can lead to uncoordinated movement. That is true, it can. Because remember, it's an insulator and you've got node-to-node jumping. But like, if we jump from one bit to the next node and there's insulation between them, if we take that insulation away, then you could get depolarization going the wrong ways as well. Because it's not going across the entire length, so there'll be depolarization here and here. So this part might not be in the refractory period, so it could go backwards like that and be uncoordinated. So that one is correct, so that one's not possible. Next one says, so saltatory conduction increases the rate of propagation. Well, it does, because it means it jumps from node to node rather than just across the entire neuron, so that one's out. The myelin sheath formed is from Schwann cells. That is true, it does. The nodes of Ranvier act as electrical impulses. No, the bits between each of the myelin bits that it jumps from one to the next are the nodes. Those are bits of exposed non-myelinated membrane, which are conductive. So by that logic, this is D. So now it says, which of the following statements are correct? The autonomic nervous system contains sensory and motor neurons. Somatic and parasympathetic motor neurons use different neurotransmitters. The autonomic nervous system contains sensory and motor neurons. That is correct, because, for example, if you want to detect like low or high blood pressure, you're going to need a sensory neuron to detect that, and also to send out impulses to do something with the motor. With the muscles, like cardiac muscle, needs a motor neuron, so that one is true. Somatic and parasympathetic motor neurons use different neurotransmitters. That is incorrect. They both use acetylcholine, so that one's out. Somatic motor neurons stimulate skeletal muscles, whereas autonomic stimulates only glands. That is not correct, because you can stimulate cardiac muscle with it. So by that, it has to be D. Next it says, NAD, FAD, and coenzyme A are molecules involved in cellular respiration. Which of the following statements about these molecules are correct? So NAD and FAD are coenzymes. That is indeed correct. Next it says, NAD is reduced by accepting hydrogen atoms. That is indeed correct. CoA delivers the 3 carbon atoms of pyruvate to the Krebs cycle in the form of an acetyl group. That is incorrect. It does not deliver 3. It becomes acetyl CoA. In the link reaction, what happens is your pyruvate is decarboxylated, so you're losing a CO2 already. So I don't even have to go further than that. I know it's got to be 2 carbons instead, so that means that only 1 and 2 are correct, which is B. So next it says, a respirometer is shown below can investigate respiration. A student placed 5 cm cubed of potassium hydroxide on the left-hand side of the respirometer. So in here there's KOH, fine. And then they suspended a basket above the liquid and placed 10 grams of respiring seed in the basket. So here we go, and there's our seeds and they're respiring. Which of the following statements about this investigation is not correct? Fine. So remember, potassium hydroxide will absorb CO2. So if these lot are respiring, what's going to happen is you're going to take in oxygen, they're going to output CO2, but it's going to be absorbed immediately by the KOH. So that means this fluid level is going to go up like this because there's a net loss of air in here. So the level of the liquid in the left-hand arm of the capillary tube would decrease, which would with time. So capillary tube containing color liquid, it would decrease. Yeah, so that is definitely correct. Then it's that's it. Sorry, whoops. There you go. Yeah. I thought it was one of those, which ones, 1, 2, or 3, whoops. So now it says, water is transported across the route by more than one pathway. Which of the following statements about water molecules moving by the symplastic pathway is not correct? So remember, the symplast pathway is where you can go from cell to cell, from the cytoplasm from one to the next through plasmodesmata and the cell walls, and that does it by osmosis, whereas the apoplast pathway is through non-living cells. So it's just where the cell walls can just have the water diffuse through them freely. So water molecules can move from cell to cell without crossing membrane. Well, that is the symplast pathway, and that is correct. Then water molecules can pass through the Casparian strip. That is not correct. Water molecules cannot. It's a waxy layer that prevents things from moving through, so it means that it forces water to go through the symplast pathway instead. So that one is true. So B. Water molecules must pass through the endodermis. No, because you can have endodermis cells with the waxy strip, which is that Casparian strip I just said, which blocks it, so that's out. Water molecules travel between cells down a water potential gradient. Well, that is true. Because it's symplast, it's between cells, so it is B. Okay, so figure 16.1 shows a drawing of a dissected human heart. Identify the structures labeled A to E on this figure. The amount of times I've had beef with people over how the heart works, it actually is it's just enough. I've had enough. right? The right and left atria contract simultaneously. There's a little bit of a gap, and then the left and right ventricles contract simultaneously at the same time. The way you can figure this out is obviously you can just look at it and say, well, left and right and whatever is the opposite, but you can also tell that the thicker wall is here, so that's got to be your left, that's got to be your right. So here it's pointing at this thing in the right top part, right atrium, that's got all these bits and bobs and these nervy bits sticking out. So that is your sinoatrial node. B has to be your right atrium. I'm lazy, I'm just going to write RA. You obviously have to write the whole thing. C, where's C pointing to? So C is pointing to something that goes into the right atrium. So venous blood from the rest of the body enters the right atrium, so that has to be the vena cava, or if we're going to be technical, the inferior vena cava. So here, D, you can see that's a valve, and it's between a ventricle and a blood vessel and an artery, so it's got to be a semilunar valve. And if you want to get technical, it's left ventricle to the aorta, so it's the aortic valve. And then E is pointing at what? So E is pointing at something between the left atrium and left ventricle, so it's got to be a valve. And it's you can either call it mitral valve, or you could call it the bicuspid valve, or the left AV valve. Either is fine. I'm going to call it mitral because that's advantage. But either one of them is fine. So then it says, state which subdivision of the peripheral nervous system supplies the structures in figure 16.1. So the central nervous system, if you think about it, is your brain and spinal cord, right? The entire nervous system split into that, or anything that isn't in the brain or spinal cord is your peripheral nervous system. So that's every single nerve outside of these two. This is then split into somatic, and then your autonomic nervous system, which is then further split into the parasympathetic nervous system and the sympathetic nervous system. So fight or flight, rest and digest. Somatic is all to do with, for example, conscious control, so skeletal muscle, like contracting and picking things up or whatever. This is fight or flight. Sorry, that's rest and digest, that's fight or flight. Autonomic is all to do with automatic stuff that you don't think about, so for example, digestion. These have opposite effects on the body, and they're always active to balance each other out and maintain a normal. So it's not like, if you had sympathetic switched off entirely, you'd only have parasympathetic, which would lower your heart rate and you'd be dead, so it's not that. So here it says which one of the peripheral nervous systems, so we're looking at this division, supplies A, which is a sinoatrial node. So it's whichever one is controlling the flow of heart, like the rate of the sinoatrial node firing, the control of heart rate. Now that's not a conscious thing. You don't have to be like, oh, I've got to speed up my heart rate, so it's autonomic. So here it's a diagram of the human brain, and it says identify the letter and name of the structure that is connected to nerves, connected by nerves to structure A. So we're looking for what the letter is. So the thing that's connected to it is the medulla oblongata, which is at the bottom of the brainstem, which is I. If you were to label all the rest of them, J is the cerebellum, which is all to do with muscle coordination, balance, and posture. If we were to look at H, which H is pointing at the pituitary gland, so that releases a lot of different hormones. And then if we look at where G is, the G is the hypothalamus, which is where it's involved in controlling body temperature, and it also produces hormones that then act on the pituitary gland. And then if we look at F as well, that's just your cerebrum, so that's where, you're thinking about, that's where you take in things and think about and actually use stuff, so like seeing, hearing, thinking, processing, vision, memories, all that kind of stuff. Normal human resting heart rate is approximately 70 beats per minute. Cutting the parasympathetic nerve increases this to around 100 beats per minute. Suggest two conclusions that can be made by the observation of the control of the resting heart rate in humans. Well, number one is that clearly, they've told you again, if you chop it, it goes up. So that means that the parasympathetic nervous system, I'm going to write PNS for short, you write the whole thing, slows down your heart rate. Number one, right? Number two is, what other conclusion can you draw? Well, it must mean that the heart rate is controlled by the autonomic nervous system, because if you remove the parasympathetic part, now it goes back up. So surely that has to be controlled like that. Another thing you can also say, but I don't think this is really a conclusion you can draw, is that the heart rate reduces if you have parasympathetic impulse by about 30 beats per minute. Not a big fan of that one though. So it says injury to the parts of the brain labeled G and H. So G is the hypothalamus and H is the pituitary. Can lead to a range of symptoms including fatigue, weight gain, menstrual irregularities, low blood pressure or dizziness, and then increased sensitivity to cold. So outline how injuries to G and H can cause a wide range of symptoms. So, well, first of all, this is the hypothalamus and pituitary that are damaged. And, well, you need to think about what's going on. Well, it's these two produce many different hormones, which then means that other glands, like your adrenals and stuff, are affected and non-functional, and that causes symptoms. So, for example, one of the hormones you could think about is follicle stimulating hormone and luteinizing hormone as well. So that can lead to irregularities in that stuff as well. So decreased FSH and LH gives you menstrual irregularities. You could also talk about low cortisol, for example, as well, which can cause problems like that. You can talk about thyroid hormones. There's lots to talk about, but here, that's what you got to do there. Next, it says, suggest why it can be difficult for a doctor to conclude that symptoms described in part 2 are definitely caused by damage to part of the brain. So if we think about it, the hypothalamus releases hormones that stimulate the pituitary gland, which then goes and stimulates your endocrine organs. So for example, that could be your ovaries, it could be your testes, it could be your thyroid, it could be your adrenal glands. So damage to any one of these can cause problems. So that's the issue, is that you could damage, for example, like your thyroid, and then you don't know, without doing some tests anyway, whether the thyroid is the problem or one of these. So that's what it is that damage to endocrine organs can produce similar symptoms. That being said, though, we have ways of testing it. So, for example, with thyroid disease, we can test thyroxine, which is where the T3 and T4, which comes from the endocrine gland, the thyroid itself. But we can also measure thyroid stimulating hormone, which comes from the pituitary gland. So if there's high TSH, it means that there's no thyroid hormones being made, so it can't put negative feedback back on this. So it's just showing you that these things sort of like interlink. So complete the following passage using the most appropriate terms. So the knee-jerk reflex is when what happens is you have stretch of the quadriceps muscle, and immediately it straightens your leg, and this is to help you sort of keep your balance and posture. So what happens is that you have stretch receptors in the quadriceps muscle detecting that it's stretched. An impulse is travels across a sensory neuron, but then it goes into your spinal cord, and it, rather than going through relay neuron and this and that blah blah blah, it actually communicates straight away with a motor neuron, and it comes right back out. So there's no relay neuron here, and that means you don't have to have that extra delay of thinking about it. So in this case, the knee-jerk reflex is an example of spinal reflex because it only goes through the spinal cord. Whereas the blinking reflex is a cranial reflex because it goes through the brain. It wouldn't go through the spinal cord because it would have to go down from your eyes back up again. It's just pointless doing that. Reflexes are very fast as they only involve 1 or 2 synapses, which are the slowest parts of nerve transmission. This is the reason that reflexes give a survival advantage, because if you think about it, it's just action, right? That's pretty fast. So clearly, the other thing that's limiting it is whatever's between each of the action potentials, connecting sensory and motor neurons, which is the synapse, where neurotransmitters have to diffuse across, and that's what slows things down. The pancreas produces digestive enzymes and is also involved in the regulation of blood glucose concentrations. So figure 17 in the insert, which I've pasted here, shows a light micrograph of a section of mouse pancreas. Identify the structures K and L in this figure. So whenever you look at stained sections of pancreatic tissue under a light microscope, you can only really differentiate between alpha and beta cells if you've used stains to make them different colors. So already these obviously has been highlighted here. So there's characteristics that make them different. The purple stained cells are the beta cells, and the pink stained cells are alpha cells. That's just how they work. That's just how it be. So in that case, that must mean that K, well, the K is pointing at all of this stuff, remember? So if we look at this here, we've got L here and we've got K here. Now when we're doing staining of like pancreatic tissue, the beta cells and the alpha cells come up differently depending on the stain that you use, and it's usually that we use a stain that makes them like purplish. So the islets of Langerhans are lightly stained, but if we went further and looked in them more detailed, beta cells tend to be more purple and dark, whereas alpha cells are pink. However, we've got this cluster of them here. So what we're actually looking at is we are looking at the islets of Langerhans amongst all these other cells. So whenever you're looking at a tissue slide like this and it's pancreatic tissue, you've got a patch of like sort of pale stuff with intermittent, darky, whitey, pinky bits in it. That's the islets of Langerhans there. So those are the islets of Langerhans, islets of Langerhans there. So in that case, L, you should be able to recognize just because you can see it they always kind of look like this. It's a blood vessel. Scientists investigate the effect of this drug called nifedipine on the secretion of insulin from pancreas cells in culture. That's a calcium channel blocker. Pancreas cells are the first incubated with glucose and a concentration of this. The concentration of glucose is then increased in the presence or absence of nifedipine. The scientists then measured the amount of insulin secreted by the cells, and they recorded them as a percentage the percentage of the total insulin content of these cells in each experiment was repeated 7 times. So just real quick, what they did is they got cells, the pancreas cells, in glucose, and then what they did is they had 2 groups. They had nifedipine and then no nifedipine, and what they did is they increased the glucose concentration. So what we would expect to happen just on that alone is that the insulin should go up in both of them, right? Because we're increasing the glucose. Insulin secretion should increase, and glucagon secretion should drop. So then what they did is they recorded the amount of insulin secreted, and then they did it as the total percentage insulin content of the cells, meaning they took all of the insulin these cells have, and then took however much was put out, like secreted out, and then found the percentage of this of the original. Fine. So it says name the cells that secrete insulin, they're just the beta cells. It says explain why it was necessary to increase the concentration of glucose surrounding the cells before they measured insulin secretion. So they've quite literally said they must increase glucose, and now they're measuring insulin secretion. So based on that question alone, you just got to think, what are the gaps? Well, I mean, glucose, like increased glucose increases insulin secretion, right? So then we need to think why, we need to explain why that means that, that helps measuring insulin secretion. Well, it means that it would lead, increasing glucose would lead to a change in insulin secretion. Or you could say, for example, I don't know, you could say maybe it could be high enough to be measured, but the whole point is that they're increasing their sugar so they can see there's a difference, it's going up. So now it says suggest and explain which statistical test they will use to analyze their data. So we're looking at, this is the conditions of the cell without nifedipine, with nifedipine, so they're two separate groups. And then it's not like the cells, there's like one and then it's going through the next thing and then the next thing. It is two separate groups that are going through these things. And they've measured the mean insulin secreted. So we're looking to see if there's a difference between the means of two separate groups. So that means it has to be an unpaired t-test. Why? Because we are comparing the means of two different groups. The statistical test gave a value of p being less than 0.001, using the words chance and probability to draw a conclusion from the statistical test. So remember that this is talking about the differences in means. So p is less than 0.001, meaning that this is a significant result, as in this p-value is significant. But you can't just say the results are significant. You need to say what this p-value is testing. This p-value is only testing whether there is a difference between these two. That is all. It's not testing anything else, and you can't talk about anything else. So therefore, it means that the probability of, and then don't say just the results, say what they are. The difference is, or the difference, sorry, I should say, difference in mean insulin secretion being due to chance is less than 0.001. There you go. So therefore the results are unlikely to be due to chance. So now it says nifedipine blocks calcium 2+ channels. Explain how blocking calcium channels could inhibit insulin secretion. So we need to think about what could happen here. I'll just do a quick crash course of a beta cell and what happens with them. So when glucose concentration is high in the blood, you get more glucose entering via facilitated diffusion. When you have more glucose in a beta cell, you've got more respiration, and then you get more ATP. But then, if you have more ATP happening, then K+ ion channels close. So that means potassium ions can't get in or out. So then what happens is you get this buildup of K+ in the cells. So the inside is comparatively more positive versus the outside, or less negative. So then what happens is you've got this depolarization of this beta cell. What happens is this depolarization triggers calcium ion channels to open, so that calcium ion channels move into the cell via facilitated diffusion. Calcium ion channels moving into the cell means that vesicles containing insulin fuse with the plasma cell membrane, and then it leaves via exocytosis and the insulin is released, similar to how neurotransmitters work. So in this case, if we block calcium channels, it means that Ca2+ doesn't enter beta cells, therefore you get less exocytosis of insulin, and therefore you get less secretion. So type 1 diabetes has been treatable for many years, but the treatment's always improving. Evaluate the treatments of type 1 diabetes that have been used in the past and the potential and future treatments. What we need to do is, it's saying evaluate, so we need to talk about the advantages and the limitations, and it's talking about what's used in the past and the potential and the future stuff. So if we look at the past, remember type 1 diabetes is where you're not making any insulin because you have autoimmune destruction of the beta cells. So the way we treated it in the past was by just giving them insulin. So we used to treat with insulin from, and it used to be from animal, like pigs and stuff. Now, the advantages of that is that, I mean, well, it meant that people wouldn't die. That's a pretty good one. Wouldn't die, because we didn't have any other stuff, and also, I mean, it was relatively safe, because we tested it, and it'd been fine, right? The disadvantages, though, of using animal derives, well, first of all, is that the way we got it is we literally, had to get pancreases from animals and, break it up and separate it, so it needed purifying. As well as that, some people unfortunately had allergic reactions as well, so allergic reaction risk. It's also expensive, and also if you think about it as well from another perspective, there are ethical issues as well with just killing animals for the sake of getting insulin. So that's all the things I can think of rinsing what we did in the past. Current treatments for insulin involve so what we do now is we use genetically modified recombinant bacteria. What they do is that they can produce the insulin instead of extracting it from animal pancreases, and that's quite a good thing actually, because it means we don't have to do all of that jazz. So what are the advantages of that? First of all, some of the advantages is that it's way more pure. Like, you don't have to separate it from all these other random enzymes into, bits of tissue, this and that, glucagon and stuff from animal pancreases. It's way more pure. All you got to do is just get the bacteria out. The risk of allergy is lower because it's just standard insulin without any other stuff stuck in there. It also is cheaper. What else can I think of? And as well as that, you can just look. Now that I've like written all of these out, I can kind of just look at them and compare them too. So there's fewer ethical issues as well because you're not, killing animals, right? So like, if you do it step by step, then you can also refer back to these and like sort of jump back and be like, oh well, there's this. As well as that, what else can we do? Well, I mean, we can also like use, you can use pumps and pens with different types of long or short acting insulins too, which is quite good because that means we can have, we can customize it to behave how we want. Now, the disadvantages, though, is that, well, first of all, I mean, you can have, you're using injections, which can carry side effects. You can have skin hardening, known as lipodermatosclerosis, and that can mean it's less effective if you keep using the same site. As well as that, it can be uncomfortable. The other thing is that, you would have previous animal insulin use needed to be, like, people using that before needed to change over, which means they probably could have lost control of it. It also requires people to understand their treatment plan. So what that means is they'll have to take this insulin and then this, and they'll have to do this and that. So it's like a bit like, okay, right, fair enough. Dang. So if we look at our future / potential options, we could do islet transplants. So its benefits and its disadvantages. Well, its advantages of using islet transplants could be, well, obviously there's no need for injections. Again, I'm just comparing it to the ones above. As well as that, it means you can get less side effects as well. So for example, like hypoglycemia and stuff like that. As well as that, it can also control glucose more similarly to natural, rather than having to sort of do it yourself manually. Now the disadvantages is that, first of all, I mean, you'd need to use immunosuppressive drugs. As well as that, or you could get rejection. As well as that, if we're using for example, we need to rely on available donor tissue. What the problems are there with transplants, and I think that's pretty much that. And then, and then if we look at the other options as well, so that's transplanting the actual cells. We could transplant stem cells. So I'll just verbally talk about that. When we're transplanting stem cells, the other problem, though, is that there's an increased risk of cancer when using these. As well as that, it's expensive. Other options as well, so we could try immunotherapies, but the problem with that is that it's still in clinical trials, and we don't really have very much on that either. So those are the things that I'd say. Okay, so the liver is an organ with a wide variety of functions. Figure 18.1, which I've got here, shows a light micrograph of a section of a normal human liver. Identify what N and O are. In your liver, you need to be able to recognize stuff like in tissues. So in these parts here, when you've got that like sort of whitish, large circular bit, that is your central vein. And if we look at what O is pointing at one of these tiny little, thingy-mabobs. And here N is pointing at that gap. So that M here, those white spaces, are sinus. And O is pointing at a cell, so that's just a hepatocyte. So sorry, it's the next day, and I've been a bit sick, so sorry if I sound really, really dull and snot. So now it says the walls of the structures labeled M in figure 18.1 aligned with cells. Suggest and explain how these cells are adapted to carrying out their function. Just a quick diagram of the liver lobule. So your liver's made of lots and lots of these lobules. There's a central vein in the middle. You've got these hepatocytes sort of like sticking out and radiating out from the center. Each of these have the central vein here, which is connected to the hepatic vein, which we'll get to later. You also have lots of branches of like the hepatic artery and stuff like that, and the hepatic portal vein and the bile duct all feeding into this sort of thing as well, and they're connected at each lobule. So if I then took a slice of this and sliced it like sort of like this, so it's like in half. I've got my central vein sort of here, and then what's feeding into it is you've got Kupffer cells lining the blood vessel parts. So we've got a branch of the hepatic artery coming in this way. And then we've got a branch of the hepatic portal vein coming in this way. And then at the bottommost part, you've got hepatocytes and you've got a branch of the bile duct coming out. It's producing bile and stuff like that. So these are Kupffer cells, and these are those are your hepatocytes. Here, these gaps are the sinusoids. The sinusoids are where blood runs through them, and what it does, it will go past hepatocytes and all these other kinds of cells as well, and it will remove harmful things like, for example, extra like ammonia and amine groups and stuff like that, and also uses oxygen from the blood as well. So the blood runs towards that central vein, and then what they do is that all of these lobules of central veins connect up to form the hepatic vein. So then, as well as that, those Kupffer cells, what they do is that they remove bacteria and break down old red blood cells as well in the liver. So it says the walls of the label structured M, so this is the sinusoids, and the sinusoids are lined with those Kupffer cells and those hepatocytes. And it's told you as well that they are adapted to carry out their function, so their function is to remove harmful substances. The way that we remove and move around things in the body is nearly always things to do with light diffusion and stuff like that. So one thing you could go ahead and say is that the cells are thin cells, so there's a short diffusion pathway. Additionally, you could say that capillary walls and things like that, and these little cells on these little levels are fenestrated, which means they've got little gaps, and that means you can have permeability. To fenestre, like defenestrate, is to throw somebody out the window. So fenestrated means it's got windows in it, like little holes. So now it says, liver is a rich source of the enzyme catalase. The table shows examples of 3 terms from the list below. Complete the table by choosing the term from the table that matches each example. So if we just have a look at what catalase is a quaternary protein, and it has 4 different heme prosthetic groups. These prosthetic groups let catalase break down hydrogen peroxide and actually let it interact with that. Now, the purpose of catalase is to remove hydrogen peroxide, because that can be commonly made in a lot of metabolic reactions, and it's damaging to cells, so if it accumulates. So complete the table by choosing a term that matches each one of these. So prosthetic group is where you've got something stuck to another thing as well. So that is the heme molecule is bound to each polypeptide. So that's prosthetic group. Binding of the hydrogen peroxide substrate to the catalase causes a change in the shape of the active site of the catalase. So what that means is, remember that your enzyme's changing shape to allow the substrate to fit. So that's going to be induced fit. It's not going to be non-competitive inhibition, because the questions, like, this is what it normally works with. It's not catalase normally works with hydrogen peroxide, so it's not that it's binding somewhere else and changing somewhere else's active site. Hydrogen peroxide binds to the active site of this catalase, so it has to be induced fit. Cyanide ions decrease the action of catalase even at high concentrations of hydrogen peroxide. So you've got catalase, even if you have all the hydrogen peroxide in the universe, it's for some reason not working. So clearly, even if you have like a non-competitive inhibitor or competitive, it's clearly, even despite the fact that there's more of this eventually colliding with the enzyme, there seems to be no action. So what's probably happened is there's non-competitive inhibition, making the active site change shape, and it's no longer complementary and so forth. So that one has to be non-competitive inhibition. So figure 18.2 shows the effect of concentration of hydrogen peroxide and the rate of reaction of catalase. As expected, just describe it to yourself real quick. The rate of reaction goes up as you add more and more hydrogen peroxide, but starts to level off because it's an enzyme and you've got too much substrate and not enough active sites. So now it says cyanide ions inhibit catalase even at high concentrations. Sketch a line on figure 18.2 to show the effect of repeating this experiment with cyanide ions. So it's told you that even if you add a really, really high concentration, it inhibits it. So this is a non-competitive inhibitor. So non-competitive inhibitors change the active site, like I was just saying before in the previous question. So it's going to actually reach a lower highest rate, even if you keep maxing it out, because those bind and change the active site and so forth. So what happens is the reaction increases slower and more slowly compared to the original, but levels off at a lower point, because all of those inhibitors have now bound to the catalase and change its active site shape. So no matter how much you add hydrogen peroxide, it can only go as far as this, because there's fewer active sites in as a whole, which is why it levels off lower down. The Hill reaction is a model system used to study the light-dependent stage of photosynthesis. It uses a blue dye DC pip, which goes colorless when it's reduced. So I'm just going to squiggle that down. So DC pip is blue to colorless when it's reduced. Fine. So just have a quick second to understand what's going on here. So we've got supernatant, pellet, and distilled water. So pellet would have all your chloroplasts and stuff like that. So you would expect there to be the light-dependent reaction happening, whereas a supernatant wouldn't. So you wouldn't really expect that to happen either. So what they've done is they boiled for 5 minutes here, and then they haven't. So in the ones that they have, that you would expect that everything's like denatured and dead and whatever, right? Fine. And then DC pip's been added to all of them, including the control, which would be E. And then illumination light, dark, light. So Remember, this is a light-dependent reaction, so we'd expect that to have, like, nothing. Color after 5 minutes is all blue, 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 colorless blue. Fine. So as in DC pips reduced, DC pips DC pips not reduced, not reduced, not reduced, reduced, and not reduced. So it's just really important just to have a quick think about that, because then you appreciate what they're changing. Between each tube, they're changing one factor. What is the name of the final electron acceptor in the light-dependent stage of photosynthesis? So the whole point of the light-dependent stage is you need to generate reduced NADP and ATP, because the Calvin cycle then uses it. The light-dependent reaction, in a quick summary, what you have is you have photosystem 2, photosystem 1, the electron transport chain, and you've got chlorophyll. So light shines on chlorophyll, it releases an electron. The electron now has too much energy, it's really gassed. So what it does is it moves down the electron transport chain to photosystem 1. Now the problem is that for it to be passed over to this, it has to lose energy, otherwise it just wants to stay out. It's like, yay, I'm too excited, I'm too high energy. As it moves down, it loses energy because it's too gassed. So then what happens is, once it gets to photosystem 1, after it's lost its energy, more light shines on photosystem 1, and then it's passed on to NADP, which becomes reduced NADP. But that doesn't explain where the ATP bits come from, and also now this thing's missing an electron. So what happens is light also splits water into protons and electrons. The electron gets passed on to chlorophyll, it's replaced it, and these protons are just chilling. But what happens is, the electron that's lost its energy down the electron transport chain, that energy has to go somewhere. So what it's used for is pumping protons like this into the stroma. So that electron's lost energy, remember? So what happens is that the energy that was lost from that electron is used to pump H+ ions into the stroma of the thylakoid membrane. And these protons now want to go back out. The only way they can is through ATP synthase, which is a channel protein embedded in them. So it moves through ATP synthase, and that's where you get your ATP from. So that's called non-cyclic photophosphorylation. Photophosphorylation, photo, light, phosphorylating, ATP is being made. And it's non-cyclic because that electron is being passed on to NADP and taken away to the Calvin cycle. It's not cyclic, it's not coming back. So I just know that people will have read final electron acceptor and then gone ahead and said oxygen. It's asking what's taking it from the light-dependent reaction, which is going to be NADP. It's not reduced NADP, it's not NADPH, any of that. It's what's taking the electron. So now it says DC-PIP is reduced in the Hill reaction. So Justin explained the function of DC-PIP in this Hill reaction. So you need to look back again and have a look. So we're looking at what studies the light-dependent stage of photosynthesis, and DC-PIP gets reduced. So you need to think, well, what's the only thing that can get reduced in the light-dependent reaction? It is the NADP. So what happens is, the whole point of this is that the DC-PIP acts as the final electron acceptor, and it replaces NADP as the electron acceptor. And there you go. So now it says, using the results in the table, explain what can be concluded about each tube or pair of tubes about the light-dependent stage of photosynthesis. So if we look at tube A, we've got supernatant, we've not boiled it, we've given it DCP, we've given it light, so we're DCPIP, we've given it light, but there's no reduction because it's not gone colorless. So the clear thing here is that there must be no, photosystems, because that's where all of this is happening, remember, in the supernatant. Or you could say there's no thylakoids or chloroplasts in the supernatant as well, and that means, therefore, the light-dependent reaction takes place in the chloroplasts or thylakoids, either one. Now for tube B, what they've done is they've got the pellet, that's fine, so we've got what we want, but they boiled it for 5 minutes. Everything else is ideal, but there's still no DC pips, so we've got to think now. They boiled it, and for some reason, the light-dependent reaction stopped. So clearly, the only thing that's probably the case is that enzymes are needed for the light-dependent reaction, because boiling them stopped. Boiling the tube stopped it. So it says C and D now. So we've got the pellet, we've not boiled it, we've given it DC pip, we kept one in the dark, but the other one's in the light, but everything else is ideal. And you can see there's no reduction in the dark, but there is in some light. So therefore you need light for the light-dependent reaction, which I know seems a bit pointless, but it's what you're concluding from here. And then it says for tube E, what have they done? So tube E is you've got water and everything else is ideal, so you wouldn't expect that to be reduced. So therefore DC pip does not, reduce spontaneously or randomly or something like that. The whole point is to check that DC pip is, being reduced and like becoming colorless as a result of everything else, and not something else that's making it doing it. That's the whole point of this tube. If this DC pip did, the only thing I can conclude here is DC pip's not being reduced in nothing, so it doesn't do it on its own randomly. The student knew it was important to use sucrose solution when homogenizing the leaves. Explained why it was important that the pellet was suspended in buffer solution and why it did not contain sucrose. So it's buffer solution, no sucrose. So buffers, what they do is they maintain the optimum pH, or they resist pH changes. And then why no sucrose then? Because, well, have a think what would be going on. So no sucrose here is a bit trickier because it's told you it's nothing really to do with water potentially stuff. It's told you that like it's important to use sucrose for homogenizing. So in this case, sucrose somehow must aid this process. That's what the question's told you, and that is the case. So what happens is that you can use sucrose, and that helps like sort of. So based on that principle, the sucrose, and you've also done that. You should have done this in the required practical where you use sucrose for this as well, is to prevent further damage to the chloroplasts. Now it says, suggest and explain 2 improvements that increase the validity of this method. So validity means what ways of making this so that anyone else can do this experiment again and get the same results. So you need to look through each and every single one of these steps and think, what could go wrong if I tried doing this? So what they've done is they've cut these leaves and put them in a solution. Generally, when you do this experiment, you do it in a nice cold solution. And to my knowledge, it doesn't say that here. So first of all, you've got to use ice cold solutions. Why? It's to reduce the rate of enzyme reactions. Oh, whoops. Pretend that's there. I've written it up there by accident. Then, for the second improvement, so again, let's have a look. So then they homogenize it, whatever, and then they filter it and put it in a centrifuge tube, and they centrifuge at high speeds. Well, remember that like when you're doing centrifugation, each pellet and supernatant are going to have different things in it. So you would probably want to do it at different speeds instead of just one. So centrifuge at different speeds. Why? It's so that the pellet. Oh my god, I've done it again. Pretend that's down here. So the pellet mainly has chloroplast. Otherwise, it's going to have loads of other bits and bobs in it. Especially if it's got things like mitochondria, which also involve oxidation reduction that can screw up DC pip. As well as that, let's have a look what else I can do. So, as well as that, if we look, we've resuspended it in a buffer. Again, nothing about the temperature, so we could probably, maybe put it in like a water bath or something to keep them consistent. And then as well as that, if we look, we have them in dark versus light. Well, light changes throughout the day. It could be that, it's like sunset or something. So it's probably good to just have a fixed lamp there and a set wavelength as well, which can affect all of this stuff. So those are just if you go step by step, that helps. So terrariums are popular for growing houseplants. A terrarium is a glass container containing soil and small plants. Once established, a terrarium can be sealed and the plants will be able to grow for months or even years without having to be in contact with the outside atmosphere. So just one other reason why the plants in a sealed terrarium continue to grow. So you need to think, well, what do these plants need that they would have gotten from the outside atmosphere? The only thing would be oxygen and CO2. So in this case, they must have a continued supply of oxygen and CO2. So they must either respire, which produces CO2 for photosynthesis, or they photosynthesize, which produces O2 for resp. Additionally, you could go ahead and say as well that, well, these things could fine respire and photosynthesize, but why are they still able to die and things are able to grow back? Well, you could also have decomposition as well, replacing nutrients and returning them to the soil so they can be used again. So cacti are popular houseplants, suggest why they do not grow well in a terrarium. So the key fact that's interesting about cacti is that they are xerophytes. And the question has told you that it keeps them moist as well. So the reason that they probably don't survive is because it's just too moist. So it's working basically with what the question says. The figure shows a pitometer used to measure the rate of transpiration in a leafy shoot. So you've got a leafy shoot, you've got rubber tubing, water reservoir, screw clip, capillary tube, and water meniscus. So if it's transpiring, what it means is that water is escaping through the stomata, so water leaves like this. So then you would expect that to go that way. So besides safety precautions, explain one practical precaution that should be taken when using a pitometer and explain why. So let's have a look what's going on. So precaution practically means we need to look at what could go wrong with this that would stop this from happening. So we want to make sure that this transpiration into water is leaving and this is getting dragged that way, that little water meniscus. So we need to make sure that this is entirely perfectly sealed. So we need to prevent air entering you know, at like the rubber chute, or for example where the cut end is, the rubber end, sorry, rubber tubing end. And Y, it ensures water is a continuous column, so it means it just gets dragged across. Also, you could say something like, for example, you could keep temperature the same, which, because that means you know, higher or lower ones can affect the rate of transpiration. And what else could you do? You'd also want to make sure that's closed while you're running it as well, because if that's open, water will just come back in here and it'll just balance everything out again. So now it says, a student carried out an investigation to measure the effect of air movement on the rate of transpiration. What they did is they used the apparatus, added an electric fan to make air movement. They measured the diameter travelled in 5 minutes, and then they replicated it 5 minutes with the fan switched off, and then 5 minutes with it on, and here's the results. So they've repeated it, and then they've got a mean, and they've got a standard deviation, and that's fine, whatever. And it says, calculate the standard deviation for this distance travelled when the fan is switched on. Use the formula S equals sigma x minus x squared over n minus 1. Fine. So S is standard deviation, sigma means the sum of, n is like the number of the values that you have, and then x is like a data point, like a value, and then x bar is the mean. So if we're going to work out what that, if I call that, that's going to be x, I want to work out x minus x bar all squared. So we're going to do that for each of them. So what we need to do is we do 128 minus 125.6 and then square it. So 128 minus 125.6 squared, it gives you 5.76, and then you rinse and repeat for all of them. So the next one would be 124 minus the same thing, so that would be 2.56, and then it would be 125 minus that, which is 0.36, and then 123 minus that, which is 6.76, and then it would be 128 minus that, which is 5.76, and there. So then all we need to do is we need to work out sigma x minus x bar squared, so that would be the sum of all of that, which is going to be 5.76 plus 2.56 plus 0.36 plus 6.76 plus 5.76, which gives us 21.2. So then S is going to equal the square root of 21.2 over the number minus 1, so it's going to be 5 minus 1. So if we work that out, we should get an answer of 2 which gives us a sum of 21.2. So then that means S is going to equal the square root of 21.2 divided by n minus 1. We have 1, 2, 3, 4, 5 replicates, so it's going to be 5 minus 1. If we do that, we should get the square root of 21.2 divided by 5 minus 1, which gives us 2.302, which is roughly 2.3. So then it says, what can you conclude from comparing the standard deviation of the two means? So the standard deviation means how much spread there is of all the results around the mean. It's not the same as the range, because for example, if I have the mean here, the mean might be that, but you could see that the range above it is like shorter than the range below. There's more values below than above. So standard deviation, what it does is it puts the mean in the middle and then shows you the range of the results around it. So that's why you can always see that it's kind of like this every time in the middle. So here, what could you conclude about comparing them? Well, this one's lower than that. So that means the fan off data is less, the fan off data, is more spread about the mean. What I mean by that is if, for example, if this is the mean there, then it would show this. So you've got lots of things above, lots of things below. Whereas here, that's quite less spread. This is a sort of better way to visualize the spread of results than just using ranges, because again, that looks like there's not much spread here, but there's a pretty hefty amount of spread there. The student carried out another investigation. They wanted to estimate the total leaf surface area of the chute, and then what they wanted to do is they wanted to remove all the leaves from the chute and then place the leaves at the ground on graph paper, count the square, and estimate the area. State two things that they need to do to make sure the leaf area is accurate. So accuracy is how close you are to the real value. So imagine if I have a bullseye. If I hit the bullseye, that's accurate. Whereas if I miss the bullseye, it's not accurate. And precision is how many times you get the same value if you repeat it. So if I'm accurate and precise, it means I hit the bullseye and I do it again and again and again. But I could be accurate the first time, but then I'm not very precise. Or I could be not accurate, but at least when I do it, I get the same results, it's precise. So in this case, they want to see how can you get to the point where it's the closest and realist value. So they're measuring the surface area of a leaf. How can we make sure that this is the true surface area of the leaf? So if you put a leaf on paper, it's probably not going to be flat. So flatten it is one of them. Or what else? You remember that some of them are going to be, the squares are going to be partially covered. So, factor in partially counted squares. As well as that, if we're looking at the surface area of a leaf is just not one side. There's the top and the bottom, and there's also the sides as well, but that's kind of hard to work out because they're so flat. So you would need to double the area to give the area of both surfaces as well. So it says, in this investigation, they calculated the rate of transpiration to give you 30 millimeters cubed per minute. That's the millimeters cubed, so you need to think that's a volume. What's that going to be? A volume of air, or volume of water, or something like that. And it's per minute. And then they estimated the total leaf surface area of this shoot to be 37 centimeters squared. Calculate the rate of transpiration in centimeters cubed per hour per centimeter squared. Give your answer in standard form to two significant figures. So what they're saying is that this means it's per centimeter squared of leaf. So what they've got is this leaf here, and they're trying to work out the transpiration of this one area, or this one, for example. So in this case, a per centimeter squared. So here, it's 30 millimeters cubed in a minute. They want it in centimeters cubed per hour. So I'm going to go for the hour part first. That's going to be 1,000, because in 1 minute we can do 30. So in an hour we do 60 lots of that. So it'll be 30 times 60, which gives you 1,800 millimeters cubed, per hour. So I know that 1 millimeter is going to equal 0.1 centimeters. The reason why is because you divide it by 10. So therefore, 1 millimeter squared, well, that's 0.01 centimeter squared. Why? Because you divide it by 10, but also divide it by 10 again, so divided by 10 squared. So 1 millimeter cubed to centimeters cubed must be divided by 10 cubed. So in this case, it means divide by 1,000. So we're going to divide that by 1,000, which would give us 1.8 centimeters cubed per hour. But then you can see they want it per centimeter squared, which means per centimeter squared of the area, which is there. So 1.8 divided by 37 gives you 0.04, and they want it to two significant figures, so we're going to go 47 and then those units, but they want it in standard form. So then what that means is we're going to have to then 0.047 change that to significant figures, which would be 4.7, but the question wants it in standard form, so it's going to be 4.7, and then how do we get back to there? So it would be times 10 to the 1, 2, times 10 to minus 3. There you go. So then it says, describe how new xylem cells are produced. So cells in the meristems are what differentiate into xylem and phloem. The stem cells that are doing that, the stems that are doing that are called stem cells. So there's stem cells in the meristems, and if you want to be more specific, it's vascular cambium, and then what they do is they differentiate. Explain how glucose produced in photosynthesis is translocated to parts of the plant where glucose is metabolized or stored. So it's not asking you to describe photosynthesis, it's saying how is the glucose made, moved to different parts where it's either metabolized or stored. So what it's asking you to describe is translocation, which is where you move dissolved substances to where they're needed. If I diagrammatically draw what's going on, so we've got our phloem and all of our bits and bobs going on in it, like so. Cool. And then we have like what we call the sink, which is like a cell where it's needed, and then we've got our source, which is where the cell is made, and we've got like this will be our companion cell. This is our phloem. I'm going to put the xylem in here as well as a really badly drawn thing here, just for the sake of diagrammatic purposes, and I'm going to make this red dot the sucrose. So what happens is that active transport puts loads the sucrose into sieve tubes in the phloem at the source. So it puts it into here like this, it pumps them in there. So in its first step, what happens is the companion cell loads sucrose into the phloem. The reason they use sucrose is because it's less likely to be metabolized as it goes down its journey to the other parts, whereas if it was just glucose, it'd probably get used along the way. So if I zoom in on what's happening with this companion cell to do that, the companion cell it actively transports H+ protons into the environment around it. What then happens is you've got a higher concentration outside than inside the cell. So what it wants to do is it wants to get back in the cell, but the only way it can is by binding to a co-transporter protein. This co-transporter protein will only open if both things are bound to it, and the other thing that binds to it, lo and behold, is sucrose. So then what happens is sucrose enters the companion cell. Now you've got a high concentration of sucrose, and I should have drawn them, but you've got plasmodesmata that link this to the sieve tube. So now there's more sucrose in the companion cell than there is in the phloem. It moves out into the phloem through the plasmodesmata through from facilitated diffusion. So that's how it works. The first part is to just shove as much sucrose as it possibly can into the cell by using that co-transporter, and then the concentration gradient is higher of sucrose than in here. The active transporty part is the proton itself, really. So then what happens is now that we've moved the sucrose into the phloem, there's going to be a drop in the water potential. So what happens next is that the xylem, water moves via osmosis from the xylem into the phloem. So now you've got more water than there should be in the phloem. There's a bigger volume of stuff going on here. So what happens is the pressure in here gets higher. Now pressure likes to move from a higher to a lower pressure, like down a gradient. So for example, if you squeeze a water bottle and the lid is off, it comes out of the bottle because the pressure in the bottle is higher than it is on the outside of the atmosphere. And that makes sense because if you were a tiny person in that water bottle, you'd be squeezed from all over, and that's high pressure. And outside, we're not doing that because it's only air particles that are putting pressure on us. So then in that case, what happens is all of this stuff moves down the phloem, down a pressure gradient, including the sucrose and the water and so forth like this, until it gets to the sink where it is needed. So at the sink end, the concentration of sucrose is higher than in the sink. So what happens is it can actually just move in by facilitated diffusion, but you could also have active transport as well if you really want to try and get it all going. Then, once the sucrose is back in here, the water potential in the phloem now goes back up because there's less dissolved sucrose in it. So then what happens is this water then moves out, back into the xylem, down via osmosis, down a water potential gradient, and then what happens is that this sink then either uses or stores that sucrose. The reason being is because the moment that's either metabolized into something else, or respired, or stored, it re-lowers that concentration, so the concentration is always higher than higher above than it is here, meaning there's always a concentration gradient maintained. So now to turn that into words, well, first of all, again, we're not going to use glucose. The question's asking you how glucose eventually does all of this. It's sucrose. So glucose is converted to sucrose. So what I described there was the apoplast route. Then what happens is you've got active, sorry, and then I should say it's loaded into phloem by apoplast route. So active transport of H plus from companion cells into surroundings. Then H plus moves into cell again, down the concentration gradient with sucrose via a co-transporter protein. I should add that requires ATP as well because it's active transport, remember. So then what happens is the sucrose can then be loaded in via the symplast route through plasmodesmata. So sucrose enters phloem passively via plasmodesmata, which is your symplast route. And then what happens is this lowers the water potential in the phloem. So then you get water entering the phloem from the xylem down a water potential gradient. Then what happens is that increases the hydrostatic pressure. So then what happens is the substances all move down phloem, or up, whichever way, I should say. It moves through the phloem to the sink, down a pressure gradient. Then what happens is that it goes from source to sink. In this case, have they told us what it is? Oh, it's just, in general, it's a leaf, but it could be whatever. And then sucrose diffuses into the sink cells. And then it's asking you to describe where it is metabolized or stored. So remember, the glucose is now sucrose. We've got to change it back into glucose because the question's asked us to do that. So what happens then is that it's converted into glucose, and then it's either metabolized, it's used in respiration, or it's converted to starch for storage. And again, doing that maintains that concentration gradient, like I said before. So this maintains concentration gradient. And there you go. That's how you do it. So the formation of tissue fluid is an example of ultrafiltration. Osmosis plays an important part in this. The water potential of the blood depends on the concentration of solutes such as glucose, amino acids, and mineral ions, as well as large plasma proteins. State the effect of the water potential on the blood if the concentration of glucose increases. A lot of people read this and think it's something to do with tissue fluid. It's literally just saying what happens, water potential, if glucose high. So the water potential decreases. Now it says, explain why the oncotic pressure of blood depends only on the concentration of large plasma proteins. To understand this concept, we'll just do a quick run through of what tissue fluid is. So you've got your arterial end here, which is relatively high pressure, and eventually it branches out into capillaries, and then you've got your venial end as well. So arterial end, the venial end. So what happens here is you've got a relatively high pressure and then you go into a low pressure here. So for example, if you have like this big pipe, right, and then there's water flowing into here like that, and this pipe's got like huge amounts of volume of water. Imagine these next pipes have tiny little holes in it. Because the pressure like is now even higher because the volume's now dropped, all of the stuff squeezes out like this, and that's exactly what's happening here. The arteriole is a big pipe, and then the capillaries are little pipes with little holes in them. So what happens is things that can fit through the little holes can get out. So in the arterial end, what happens is water and little solutes can move out, but big things like proteins and blood cells don't stay in here. Then what happens is that forms fluid around the cells and tissues, but then we don't like that doesn't always just stay there, because at the venial end, remember that as water is being lost at these parts, within the capillaries, the blood water potential drops. So here, the water potential is quite low because a lot of it's been shoved out, so it actually moves back in via osmosis to here to balance things out, and that's your tissue fluid. Now, that's one way of sort of controlling, the amount of tissue fluid forming and stuff as well. However, like I was just saying there, the water moving out, and the water potential dropping here isn't because the water's moving out, it's because what's still left inside. Inside these capillaries, you've got these big chunky proteins. They're plasma proteins, and because they're still here and they can't get out, that's what makes the water potential drop. Because they're losing water, but there's still more solute. So then, at the venial end, water moves back in via osmosis as a result of these. If we didn't have any of these plasma proteins, then water would move out, and you'd have water potential depends on the amount of solute there is, not really how much water there is, if that makes sense. So if there was like nothing in here and then water moves in and out, well, I mean, the water potential is still the same, but the moment these proteins are in here, the water potential's dropped here. So that's what's happening here. Explain why it only depends on these large plasma proteins. These large proteins cannot leave the capillaries, but water can. So then you have these remain in the capillaries when water leaves, so that drops the water potential, and that's what gives you something called oncotic pressure, meaning that the water potential is lower here, so water has a tendency to move back in here again because of those proteins. So now it says the table below compares a capillary with the surrounding tissue fluids, oncotic pressure, hydrostatic pressure, and the concentration of albumin, which is a protein. So you can see here that the pressures are relatively high and the tissue fluid's quite low, and there's less albumin and there's more in the tissue fluid than there is in the capillaries. So net movement of fluid between the capillary and tissue fluid depends on this driving force, JV equals PC minus PI minus all this other stuff basically. So then it's all of these things, right? And it says the reflectance factor is a measure of how permeable the capillary is to albumin, and it varies between 0, which is totally permeable, and 1, that is totally impermeable. Inflammation can reduce the value of the reflectance factor. In one type of capillary, the reflectance factor was found to be 0.75. Use this information and in the table to calculate the value of the net driving force and predict the direction of movement. So the net movement of fluid depends on the driving force, JV. So JV equals the PC, which they've told you is hydrostatic pressure in the capillary. So that's what, 4.5 minus the hydrostatic pressure in the tissue fluid, which is 0.15, minus the reflectance factor, which is 0.75 times the oncotic pressure of the capillary minus the tissue fluid. So that's going to be 4.2 minus 0. We should get 4.5 minus 0.15 minus 0.7, 1.221, and then obviously try to follow the number of significant figures so they give you in the question, so 1.22 kilopascals. So but it says predict the direction of the movement. So it's told you here the net movement between the capillary and tissue fluid depends on the net driving force. So this is JV is between capillary and tissue fluid. Tissue fluid, capillary, meaning either that way. So if it's positive, it means it's this way. If it's negative, it's got to be not from the capillary to tissue fluid, so probably going to be down into it. So in this case, this means it is out of the capillary. So then it says nephrotic syndrome is a kidney disease, so the protein is excreted in urine, and then one group of people with nephrotic syndrome had a plasma albumin of this. Explain how this could affect the net driving force. There's no need to perform a calculation. So if we just look again, the JV equals the hydrostatic pressure of the capillary minus the hydrostatic pressure of the tissue fluid minus the reflectance factor, of the capillary oncotic pressure, minus the tissue fluid oncotic pressure, right? So in this case, we need to think about what the albumin's doing, which of these factors it's going to be changing. So the plasma proteins are what's responsible for oncotic pressure. So if we have less albumin, then we have less oncotic pressure. if you look at the formula here, what's changing is, well, in the capillary, sorry, I should say, because that's where it is. So that means this whole thing reduces. So as a result, what happens is that the JV would be bigger because this is going to stay the same, but that's now wholly dropped. So a medical student concluded that a patient with kidney disease and a plasma protein concentration of this would show signs of swelling, such as swollen ankles. Evaluate their conclusion. So they're saying low albumin plus kidney disease gives you ankle swelling, which they're not, entirely wrong. It's quite impressive. So there's that, right? We need to look at everything we've dealt with in the question to see what the pros and the cons are. So the things that are supportive or against it. So the things that are supportive of it, well, we know from the previous question and also the other bits and bobs, is that if they have low albumin, you'd get an increase in JV. But remember, we need to describe what that actually means. If you have an increase in JV, that means you're going to have an increase movement of fluid out of the capillary, because remember that measures the net driving force of the movement of fluid between the capillary and tissue fluid, as in out the capillary into the fluid. So that means into the tissue fluid. So therefore you get more tissue fluid, which then gives you swollen ankles because it settles at ankles due to gravity. And then what suggests that they might not be correct? Well, we need to think about possible problems with this scenario here. So we need to think about the factors that also affect all of this. So then the JV is also dependent on all of these other factors as well. So hydrostatic pressure and tissue fluid are all to do, well, we don't know, to be honest, about the hydrostatic pressure and the oncotic pressure. So no info about hydrostatic pressure, as well as that. Well, another problem is they're assuming kidney disease means ankle swelling. Well, how do we know that that's, that the fluid is like not going somewhere else? So for example, kidney disease, you could end up losing too much water. So kidney disease could mean too much water lost in urine, which would mean, much more, let me just write this first, water lost in urine, which could mean that, for example, you would have even less ankle swelling, or they could, not all of this contributes to this, if that makes sense. You could have had less of it in the body and more of it in the water, so you could have varying amounts depending on different people. As well as that, it's only a single patient, so it's not really appropriate to sort of assume that this could be the sign in every single person. Maybe some people can have different signs. Another thing you could describe as well, there's inflammation, and that reduces the reflectance value, which is measuring how how permeable the capillary is to albumin. So if it's going, so it's saying here it's totally permeable, but it's told it's reduced. So if you reduce the reflectance factor, that means you could get more albumin in the tissue fluid, which can then increase the oncotic pressure, which could actually then mean that there's less fluid moving out, because in the tissue fluid you've now got more albumin. So why would there be less water moving out via osmosis or back in, that kind of stuff between the capillary and these two? So it might actually just end up being that the tissue fluid now drops and there's less swelling because of the fact there's more albumin there. So now it says the table below contains components about the composition of lymph and tissue fluid. Put ticks in the boxes to say whether they're true or false. So lymph is similar in composition to tissue fluid but has more oxygen. That is false. Lymph is the drainage of all the extra tissue fluid that's accumulated. But remember, that's come out of an arterial end and it's around the tissues, and it's probably had all its oxygen used up, and it's put back into lymph, which is then eventually put back in the venous system, and it returns to the heart via the veins. So in that case, by definition, it's got to have less oxygen. Tissue fluid does not contain lymphocytes because they're too large to pass through the capillary wall. Tissue fluid does not contain lymphocytes because they're too large to pass through the capillary walls. That's not entirely impossible, because when you have inflammation, the capillary walls can get wider and more leaky. So that one, they can enter tissue fluid and inflammation like that, for example, if there's an infection. Next, it says lymph contains more protein than tissue fluid because of anti-production by plasma cells. Well, yes, that's because your plasma cells are found in the lymphatic system as well, so they're making lots of things like antibodies. So they're lymphocytes, lymph cells, sites. Thank you very much. I hope that helps. Please like, share, and subscribe. You know the drill.